Assalamu alaikum, this is Dr. Narmeen Haseeb when you're in class of Physiology 1. Uh, let's start with red blood cells. Bismillahirrahmanirrahim. So previously we have studied about the blood and its composition as well as the plasma proteins and their composition. Now let's see what are RBCs and how do they work. First of all, there's a little introduction about the RBCs. RBCs are the cells which have no nucleus inside them. They have no organelles inside them. Yes, that's true. And there is no mammal on Earth which have a nucleated RBC except camels. So that's a point to remember. Then the important thing is the color of RBCs. Why are RBCs red? RBCs are red because of a coloring pigment present in them, which pigment is very important. And that pigment is known as hemoglobin. So hemoglobin is responsible for the transport of respiratory gases, which are oxygen and carbon dioxide across the body. So that is a brief introduction about the RBCs. Now let's see what is the normal value of RBCs, which again is very important for you to learn as you're a doctor. And you should never forget the normal count ranges of RBCs and the WBCs. So the normal count range of RBCs is from 4 to 5.5 millions, whereas in adult males it is 5 million, whereas in adult females it is 4.5 million. So now let's move towards the morphology. Morphology of RBCs is that it is biconcave in shape. You can also say it's a disc-shaped cell, the central portion of which is thinner and the peripheries is thicker. So why is the shape of RBCs is very important? The biconcave shape of RBC is important because it is responsible for the equal and rapid diffusion of oxygen across the cell, as well as it is responsible for providing the large surface area for the purpose of absorption and removal of different substances. Again, another important point of the shape is that the RBC squeeze through the capillaries. Whenever they pass through the capillaries, they squeeze themselves because of this shape. This is only possible because of this shape. Then there is the normal size of the RBCs. The normal size of the RBCs, you can see about the diameter, the thickness, and the surface area and volume of the RBCs on your books. Please memorize these values, they are important. Let's move towards the normal structure of RBCs. Normal structure of RBCs is the cells are non-nucleated and they have no organelles. We have discussed that earlier as well. Now, if they do not have mitochondria, how do they get their energy? So they get their energy through the glycolytic process. All right. Point to remember, they get their energies through the glycolytic process. Then, what type of cytoskeleton is holding up the RBCs? The cytoskeleton that is holding up the RBCs is made up of two proteins, namely known as actin and spectrin. These proteins are further anchored by another protein known as ankyrin. So altogether, these proteins are holding up the skeleton of the RBCs. So you see, they are responsible of providing the cell its shape. But if any of the following proteins, and specifically if spectrin is absent, what will happen then? Then there is a condition known as hereditary spherocytosis. Yes, hereditary spherocytosis will occur. It will be when the RBC shape will turn from biconcave into the spherocyte or globular shape. This shape is not as much practical as the biconcave shape is, and spherocyte shape of the cell is very fragile and is responsible for the rupture of the cell. So, point to remember is hereditary spherocytosis. I'll give you a few seconds to absorb what we have studied so far. All right, now before moving further, we need to revise a few points. RBCs are non-nucleated. RBCs have no organelles in them. RBCs have a red coloring pigment hemoglobin, which is responsible for the exchange of gases across the body. Then the normal value of RBCs, the morphology of RBCs, and the cytoskeleton, which have three important proteins inside them, and the absence of spectrum will lead to hereditary spherocytosis. Okay, Ji. Now let's move towards the properties of red blood cells. The red blood cells have a very specific property known as Rayleigh formation. Rayleigh formation is a very interesting phenomena. It's just like when you have a lot of coins and you pile up them on each other. So RBCs do similar with themselves. They pile up each of the RBCs on itself, making a pile of RBCs known as Rayleigh formation. The next property is the specific gravity. The specific gravity of RBCs is 1.092 to 1.101. Then as the packed cell volume, we have studied about the packed cell volume earlier as well. The packed cell volume is also known as the hematocrit value, which is the proportion of blood occupied by the RBCs expressed in percentage. And I hope by now you have a very well memory of the percentages of the hematocrit value, which is, I assume you can remember that it is 45% of the blood and the 55% of the plasma volume. Yes, you're right. And the last property of the RBCs is the suspension stability because of the because the RBCs are uniformly suspended in the blood during the circulation. You cannot say that there are some RBCs on this part of the blood and there are some RBCs on that part of the blood. No, all our RBCs are uniformly suspended across the blood. All right, then there's an again very important point about the lifespan of RBCs. Lifespan of RBCs is about 120 days. Yes, they are they have their life of 120 days, and whenever uh, the lifespan finishes, they are destroyed in the reticuloendothelial system. So what happens when they are destroyed? Let's see what are the fate of red blood cells. You know, the older cells are very fragile, whereas the younger cells, with their biconcave shape, they are fit to move across the capillaries. But the older cells are not as fit as the younger one, and whenever they are passing through the capillaries, they cannot squeeze themselves, so they get get ruptured. And this happens specifically in the splenic capillaries. Yes, that's why the spleen is known as the graveyard of the RBCs. So I am opening up the flow chart. You can see the flow chart of fate of RBCs. This is a very important flow chart and you need to follow it. You see, when the RBCs are destroyed in the spleen, what happens to them? They have a lot of fragments. Those fragments release hemoglobin. So what happens with this hemoglobin? Hemoglobin is further composed of iron, globin, and porphyrin. These elements, iron, globin, and porphyrin, is now free. What will happen to them? The iron will combine with the apoferritin and it will make ferritin. Ferritin is an important source of iron and it will be stored and reused by the body. Whereas globin is a protein pool, so it will also be stored and reused by the body. And porphyrin is converted into bilirubin. Bilirubin is excreted out of the body. So this is how, after the destruction of RBCs, hemoglobin is being tackled. Take a moment to absorb what we have studied about the fate of RBCs. Already, what are the functions of RBCs? RBCs have very important functions, the most important function of which we have studied in the start, that it is responsible for the transport of respiratory gases. So you see, oxygen and carbon dioxide is being transported by the RBCs, and specifically with the help of hemoglobin. So 97% of the oxygen gets itself attached with the hemoglobin of the RBC, and then it moves across the body. And when it is attached with the hemoglobin, it is known as oxyhemoglobin. And it's not a chemical reaction. The combination of oxygen and hemoglobin is not at all a chemical reaction. It's just a combination, it's just a partnership which is being done in order to move across the body. And similarly, the carbon dioxide, 30% of the carbon dioxide moves with the help of hemoglobin across the body, and the combination of carbon dioxide hemoglobin is known as carb hemoglobin. Then the third important point is that hemoglobin functions as a good buffer. So overall, we can say that blood has a good buffering capacity. Then blood group determination. Yes, the RBCs carry out the blood group antigens, the antigen A, antigen B, and antigen O. So RBCs are responsible for the blood group determination. All right, we are done with the functions of the red blood cells. Further, we are going to study about the variations of number of red blood cells. Before moving forward, I would like to revise all of what we have studied so far. Yes, count all the important points on your fingertips. RBCs are non-nucleated. RBCs have red coloring pigment, hemoglobin inside them. RBCs is responsible for transport of respiratory gases. The normal value of RBCs ranges between 4 to 5.5 million, whereas the morphology of RBCs is that it is biconcave in shape. There are no organelles present inside them. The energy is being produced through the glycolytic pathway. The cytoskeleton is made up of the proteins actin, spectrin, and further anchored by anecrin. And then we have studied about the applied physiology of hereditary spherocytosis. Then the properties of RBCs are Reulex formation, piling up of RBCs, specific gravity, suspension stability, and packed cell volume, which is also known as the hematocrit value. The lifespan of RBCs is, yes, 120 days. And then the fate of RBCs, for which I have told you the flow chart, which is in your book, is very important. And RBC, the graveyard of RBCs is spleen. Yes, it is spleen. Then the function of RBCs are that transport of oxygen, transport of carbon dioxide, buffering capacity, and the blood group determination. All right, now we have the variations in number of RBCs. We know the normal value of RBCs, which ranges between 4 to 5.5 million. But what are the physiological variations and what are the pathological variations? What are the physiological variations in which the number of RBCs increase, and what are the pathological variations in which the number will increase or decrease. First of all, we will look forward to the physiological variations. Increase in RBC count is known as polycythemia. Yes, it is known as polycythemia. Polycythemia. Good. Polycythemia can occur physiologically when there is age. At birth, the RBC count is 8 to 10 million, but uh, during the first 10 days, because of the physiological jaundice the baby has, the number of RBCs decreases, and so on, reaching towards the puberty, the RBCs will accordingly decrease to 5 million in males and to 4.5 million in females. The next is sex. According to the puberty, sorry, before the puberty and after the menopause, the RBC's value count in females is almost similar to that of the males. But during the reproductive periods of females, the count is less than that of the males. So, according to sex, the RBC count range of females is below that of the males because of the reproductive periods. Then there's high altitude. Understanding of the high altitude is very important for you. You can see a flow chart of high altitude explanation over here as well. Please follow me with this flow chart. You can see there is a high altitude. On high altitude, there is hypoxia. And when hypoxia, all right, what is hypoxia? Hypoxia is the deficiency of oxygen or a very low amount of oxygen. So hypoxia stimulates the kidney. When it will stimulate a kidney, there is a hormone known as erythropoietin present in the kidney. It will be produced because of the stimulation of the kidney, which is done by the hypoxia. So the production of erythropoietin is responsible for the production of erythrocytes, and erythrocytes are the red blood cells. Yes, so it will lead to the high altitude, and hypoxia will lead to the production of RBCs. Then comes the muscular exercise. The muscular exercise leads to the temporary increase in the RBC count. Then comes the emotional conditions. You can see another flowchart of the emotional conditions. Emotional conditions and muscular exercise leads to the increase in sympathetic activity, and whenever the sympathetic activity will increase, it will lead to the increased secretion of adrenaline. Adrenaline is another important hormone which leads to the contraction of spleen, and the contraction of spleen will be responsible for the release of RBCs. Then whenever the environmental temperature is increased, RBCs increase in number as well. And after meals, right after you have taken your meals, because of the metabolic activities, the body needs a lot of oxygen, and for that purpose the RBCs are increased in number. So all these seven points are responsible for increasing the count of RBCs physiologically. Now, what are the points which are responsible for the decrease of RBCs physiologically? High barometric pressure deep in the sea, you will, because of the deficient oxygen tension, there the count of RBC will decrease. During sleep, there is a slight decrease in the number of RBCs, and during pregnancy as well, there is a slight decrease in RBC count because of the increase in extracellular volume. So these were the physiological variations. Now let's move towards the pathological variations. Pathological variations, obviously the increase in pathology of RBCs is also known as polycythemia. The increase will be above 7 million. There is primary polycythemia known as polycythemia vera. Polycythemia vera is when the RBC count will rise above 14 million. So you see there is a lot of difference between the normal range, which is from 4 to 5.5 million, to the abnormal range of 14 million. This usually happens in myeloproliferative disorders like malignancy of red bone marrow. Then there is secondary polycythemia. Secondary polycythemia occurs secondary to some pathological conditions such as respiratory disorders like emphysema, congenital heart disease, Ayer's disease, chronic carbon monoxide poisoning by chemicals like phosphorus and arsenic, repeated mild hemorrhages. All these conditions lead to hypoxia, and we have studied that hypoxia will stimulate the kidney will stimulate the release of erythropoietin, and erythropoietin will stimulate the process of erythropoiesis. Erythropoiesis will produce the erythrocytes in return. Erythrocytes are the RBCs. Okay. Anemia. Anemia is complete a topic which we will cover in another lecture. What are the variations in size of red blood cells? There are quite a variations in size of the red blood cells pathologically. Physiologically, there is variation between the RBCs, which are in the venous blood, which are slightly larger than the RBCs, which are in the arterial blood. But pathologically, the variations are when the RBCs are smaller, known as microcytes, when the RBCs are larger cells, known as macrocytes, and when the RBCs are with different cell sizes, anisocytes. So, when will be the pathological shape of RBCs as microcytes? It will be in iron deficiency anemia. It will be in prolonged forced breathing. It will be in increased osmotic pressure. Likewise, macrocytes will be present when there will be megaloblastic anemia. Then again, here is the term anemia, and I've told you earlier that we are going to cover this topic in another lecture. Anisocytes are present in pernicious anemia. So for right now, you just have to learn that anemia there is anemia a condition, and further it has different types which we have studied in the shapes in the different shapes of pathological shapes of RBCs. What are the variations in shape of red blood cells? Red blood cells can have a shape uh, known as crenation, spherocytosis, elliptocytosis, sickle cell, and poikilocytosis. All these shapes are unusual, and they are pathological shapes. They are not normal shapes because we have studied about the normal morphology of RBCs and its importance and advantages in the body. All right, I know it's quite a long lecture, but we are about to end this chapter and the lecture as well. What can be the variation in the structure of the red blood cells? The variation can be the punctuate basophilism. Punctuate basophilism is when there are dots, there is the presence of porphyrin. Porphyrin, yes, porphyrin is the same substance we have studied in the fate of our RBCs. So, porphyrin presence will make the presence of dots, which will show as striated appearance in the RBCs. Why this happens? This happens because of the lead poisoning. Then there is a ring in red blood cells. This ring is called as goblet ring. Why is this ring? This ring is because of the periphery of the RBCs. Sorry, on the peripheries of the RBCs, a basophilic material, a ring or strands of basophilic material appears. Why this appears? This kind of ring or twisted strands appearance occurs in certain types of anemia. Then there are whole jolly bodies. In certain types of anemia, there are nuclear fragments of ectoplasm of RBCs. These fragments are known as the whole jolly bodies. So, with this, we are done with our chapter of RBCs. I hope I am clear with all the points we have studied so far. Before signing off from this lecture, as we are in habit of revising all the topics we do in the class, so let's revise them. RBCs are non-nucleated. They have a coloring pigment known as hemoglobin. Hemoglobin is responsible for the exchange of respiratory gases across the body. The normal value of RBCs morphology of RBCs, importance of cytoskeleton, its applied physiology, which is hereditary spherocytosis, the properties of RBCs, relic formation, PCV, stability, suspension stability, specific gravity, lifespan of RBCs, fate of RBCs. The flowchart of RBC, fate of RBCs is very important. Then the functions, the transport of oxygen and carbon dioxide, the buffering capacity, which is due to hemoglobin, and the blood group determination because of the presence of antigens on the RBCs. Then the variations in number, the physiological variations and the pathological variations. The physiological variations where the increase in RBC count is known as polycythemia. It happens according to the age, according to the sex, according to the high altitude, according to the muscular exercise, emotional conditions, increase in environmental temperature, and after the meals. Whereas decrease in RBC count occurs in high barometric pressures during sleep and in pregnancy. So that was all for no, the one point we are missing over here is the pathological polycythemia. Pathological polycythemia is also known as the primary pathological polycythemia is known as the polycythemia vera, and then the secondary polycythemia. So with all this we are done with red blood cells, the basics of the red blood cells basically. And I am expecting you to listen this lecture again and again, and post down your queries if any, and please do study your books thoroughly for the complete understanding of this chapter. um, for right now I'm signing off. Thank you very much for listening me and having the patience. Allah Hafiz.